Hello, my name is Constanze Jakwerth from the Center of Allergy and Environment in Munich. Today I will present our work investigating how allergen immunotherapy reshapes the Th17-TR17 Treg axis and promotes tolerogenic immune remodeling through LT-alpha and TNF receptor 2 associated signaling pathways. Allergen immunotherapy is currently the only disease-modifying treatment for allergic airway diseases and can induce long-term immune tolerance. However, the cellular mechanisms underlying this transition from inflammatory immunity toward immune regulation are still not fully understood. In particular, it remains unclear how inflammatory Th17-associated responses are reprogrammed during the early phase of treatment. In this study, we therefore focused on the dynamic relationship between Th17 cells, regulatory T cells, and a transitional FOXP3 positive, IL-17A positive population, the TR17 cells. Using human patient samples together with high-dimensional immune profiling, flow cytometry, and single-cell transcriptomics, we investigated how allergen immunotherapy reshapes the Th17-TR17 Treg axis and promotes tolerogenic immune reprogramming. We first asked how allergen immunotherapy affects these T cell populations at the site of allergic inflammation. In both experimental airway inflammation and human allergic rhinitis samples, we observed a pronounced local accumulation of Th17, TR17, and Treg populations during allergic disease. Following allergen immunotherapy, these populations shifted back toward levels observed in non-allergic or healthy controls. Importantly, this suggests that AIT does not simply suppress immune activation globally, but rather restores a balanced mucosal immune environment. To better understand the relationship between these T cell populations, we next performed single-cell transcriptomic analysis from PBMCs from a graft-specific immunotherapy cohort and validated the findings using flow cytometry. What we found was that TR17 cells did not behave like a stable, isolated population. Instead, they occupied an intermediate transcriptional state between inflammatory Th17 cells and induced regulatory T cells. Before treatment, which you can see here on the left-hand side, TR17 cells clustered much closer to the pro-inflammatory Th17 compartment. However, after 1 year of allergen immunotherapy, their trajectory shifted toward a more central position between Th17 cells and induced Tregs. This suggests that TR17 cells may represent a transitional population involved in the establishment of immune tolerance during AIT. We then asked which signaling pathways might drive this tolerogenic transition. To address this, we analyzed cell-cell communication networks before and after allergen immunotherapy. Following treatment, TR17 cells became substantially more connected within the overall signaling network. At the same time, LT-alpha and TNF family signaling pathways emerged as dominant interaction axes, particularly the LT-alpha TNF receptor 2 signaling. Importantly, these computational findings were subsequently validated experimentally by flow cytometry, where we observed enhanced LT-alpha associated TNF receptor 2 signaling across TR17 and Treg associated compartments after 1 year of treatment, with partial dependence on TNF receptor 2 mediated signaling. Together, these findings suggest that allergen immunotherapy actively reshapes intercellular communication toward a more regulatory immune state. Taken together, our study supports a model in which allergen immunotherapy restores the mucosal immune equilibrium through coordinated remodeling of the Th17-TR17 Treg axis. In this process, TR17 cells appear to function as a transitional population, linking inflammatory and regulatory T cell states. At the same time, LT-alpha and TNF receptor 2 associated signaling pathways emerge as potential mediators of this tolerogenic reprogramming. Overall, these findings provide a new mechanistic insight into how immune tolerance develops during allergen immunotherapy and may help guide future strategies aimed at promoting more durable therapeutic tolerance in allergic airway diseases. Thank you.